friends. This video is about the mechanism of the DNA replication in the prokaryote. So as we have discussed in the previous video about their factors and the enzymes involved in the DNA replication, overall view of the DNA replication in prokaryotes. And in this video, we are just going to deal their mechanism. So it involves three stages, that is initiation, elongation, and termination. In the initiation, formation of the pre-primary complex at or IC. Or IC is the region where the replication start and their Y-shaped structure form. So an additional molecule of DNA protein and a bind cooperatively forming a complex. So DNA B and DNA C protein joins the DNA protein complex and produce a replication bubble. So DNA B and C are the enzymes involved in the prokaryotic replication, as we have discussed previous. So these enzymes is ATP binding protein. It has an intrinsic ATPase activity that hydrolyzes the ATP to ATP and inactivates itself when initiation stage ends. So this figure describes the replication process. So DNA A, B, and C are the pre-priming complex which involve in the DNA replication and start to form the replication bubble. So elongation, the synthesis of the new strand is initiated by the RNA primer, which is synthesized by primase enzyme. The RNA primer is covalently extended with the nucleotide by the DNA polymerase 3, which is very important for the replication process. So DNA polymerase adds nucleotide one by one to the growing chain. It requires a free 3-OH group located on the sugar. So the addition of the nucleotide requires energy, and this energy is obtained from the nucleotide that have a 3-phosphate group attached. The breaking of the 3-phosphate releases energy in addition to ATP. There are also TTP, CTP, and GTP. So a protein called sliding clamp hold the DNA polymerase in place as continuous to add nucleotide. A sliding clamp and the clamp loader are the part of the DNA polymerase 3. So DNA proofreading. Further, the DNA polymerase 3 proofreads the sequences for the avoiding error. The multiple primer at the lagging strand are cleaved by RNase H and removed by DNA polymerase 1. So termination start by forming the complex like TER-TUS complex. So termination occurs at the TER A and TER B because the termination protein binds with the TER A and TER B region called as TUS protein. So only DNA topoisomerase 2 remain in action. It causes the both strand associate the TER-TUS complex, and finally the two different DNAs generated. So ORIC is the origin where the replication start, then replication fork is formed and replication bubble, then tertus complex is formed and determination proceed. So all about this mechanism of the prokaryotic replication. Keep watching. Thank you.